This is chapter 29, and we're going to talk about inheritance. But before we get into inheritance, I want to talk a little bit about some terminology that might be a little confusing, like what's the difference between chromosomes and DNA and genes. So a chromosome is a dense structure that's composed of tightly coiled DNA strands with their associated histones. Chromosomes are visible in the nucleus when a cell prepares to divide because they are so tightly coiled. So here we can see this structure here. This is a chromosome, and it would be tightly coiled around these structures that are called histones. So the histones would be in here, and the chromosome is tightly coiled around those. So those are called histones. And histones are just proteins. And so they allow the DNA structure to become tightly coiled. So when it's tightly coiled like that, we call it a chromosome. And it is visible under a microscope. What the chromosome is made out of is DNA. And so up here we can see the DNA molecule. The DNA molecule is made out of 2 strands of nucleic acids. Each strand of nucleic acids has nitrogenous bases that match up with the nitrogenous bases on the other nucleic acid. These nitrogenous bases include cytosine, guanine, adenine, and thymine. Cytosine is complementary to guanine, so they will always be paired. Adenine is always complementary to thymine, so they will always be paired. If the DNA molecule is not wound tightly around those histones, then it will look like fine strands in the nucleus, but you can't see these under the microscope, and they are called chromatin. These 2 nucleic acids are wound in a double helix and then held together by hydrogen bonds. So the DNA molecule actually looks more like this. And this double helix structure is what you're seeing here. A gene is a portion of the DNA strand that functions as a hereditary unit. A gene is located at a particular site on a specific chromosome and codes for a specific protein or polypeptide. Every somatic cell in your body with a nucleus contains 23 pairs of these chromosomes. One of the chromosomes in these pairs came from the sperm, and the other chromosome came from the ovum. This entire set of chromosomes make up the genotype. The 23 pairs that are in the genotype contain the genes that will determine the physical characteristics of the person. For example, what color eyes or what color hair or what color skin a person might have, or how susceptible they are to cardiovascular disease. The physical characteristics that are genetically determined are called the phenotype. So the phenotype actually results from the genotype. As an analogy, the genotype is the set of plans, and the phenotype is the finished building. Observable features like hair or eye color are called phenotypic characters, and specific character variants like brown hair, brown eyes are called phenotype traits. The 46 chromosomes carried by each human somatic cell with a nucleus occurs in pairs. These cells contain 23 pairs of chromosomes. At amphimixis, the sperm supplied the paternal member of each pair, and the ovum supplied the maternal member of each pair. These 2 members in 22 of the pairs of chromosomes are homologous chromosomes. The 2 chromosomes in these pairs have the same structure and carry the genes that affect the same traits. For example, one member contains 3 genes in a row, and the first gene determines hair color, the second eye color, and the third pigmentation of skin. If this was the chromosome that was supplied by the sperm, then the other chromosome would be supplied by the ovum, and the ovum would have the same 3 genes in a row, and the first gene would also determine hair color, the second eye color, and the third pigmentation of skin. They're homologous because they code for the same things. Although the gene from the sperm might code for brown hair, whereas the gene from the ovum might code for blonde hair. These 22 pairs are called autosomes. Most of the genes of autosomes affect body characteristics like hair color and skin pigmentation. The members of the homologous chromosomes are individually called homologs. These homologs carry the genes that affect the same traits, and the genes are in the same sequence and are located at the same position on the chromosome. The homologs have different origins. One was paternal and one was maternal, so they don't carry the same form or version of each gene. The various forms of a given gene are called alleles. If the 2 chromosomes of a homologous pair carry the same allele of a specific gene, you are homozygous for the trait affected by that gene. If you have 2 different alleles for the same gene, you are heterozygous for the trait determined by that gene. For example, if the 2 chromosomes of a homologous pair carry the allele for hair color, and both of them carry the allele for brown hair color, that person would be homozygous for the trait affected by that gene. However, if the 2 chromosomes of a homologous pair carry different alleles of a particular gene, for example, if the paternal allele coded for black hair and the maternal gene coded for blonde hair, that person would be heterozygous for hair color. The entire set of chromosomes, all 23 pairs, is called the karyotype. 22 of the pairs are the autosomes. These are the genes that affect body characteristics. The chromosomes on the 23rd pair are not truly homologous and are known as the sex chromosomes. One of their functions is to determine whether the individual is genetically male or female. Here you can see one person's entire karyotype. You see the 22 pairs, all the way up to 22. All of those are known as the autosomes. Each of the homologs, so each one of these is a homolog, you can see the different genes. So here you see this pink area here and this pink area here. That's a gene that might carry the alleles for hair color. Then you might see the gene here and the gene here, and they might carry the alleles for eye color. You can see that each one of these 22 autosomes have varying lengths and different colors, indicating they have different genes that they're coding for on each of the homologs. The 23rd pair right here, that one is considered the sex chromosomes because this is the one that will determine the sex. If the karyotype contains an X chromosome and a Y chromosome, then that person will be male. But if that person were to contain 2 X chromosomes, then that person would be genetically female. Here's a chart that maps out the human chromosomes, and this wheel indicates disorders that may occur in humans that are genetically determined. This wheel is showing which chromosomes the disorders would occur on and the specific location of the genes where the disorder originates from. So, for example, the disorder ALS, that is determined from chromosome 21, and it's showing you the exact placement of the gene. Another example would be breast cancer. So breast cancer is genetically determined from the 17th chromosome, and it's showing you the placement of the gene on that chromosome as well. So each person inherits genes from their father and genes from their mother. I'm going to now discuss some patterns of inheritance that occur on the autosomal chromosomes. In simple inheritance, the phenotype is determined by interactions between a single pair of alleles. Approximately 80% of your genotype falls within this category. In polygenic inheritance, a phenotypic trait is determined by interactions among several genes. For example, hair color. Other than blonde or red, which are recessive traits, hair color is determined by several genes. Another example would be skin color or eye color or height. We'll mention more about polygenic inheritance later in this video. Homologous chromosomes can either be homozygous or heterozygous. If a person is homozygous for a particular trait, that means that both alleles are dominant or both alleles are recessive. In either case, that allele will be expressed in the phenotype. If the homologous chromosomes are heterozygous, that means that the chromosomes contain both a dominant and a recessive allele. The dominant alleles will always be expressed over the recessive alleles. So in a heterozygous chromosome, the dominant allele will show the physical characteristic. The physical characteristics of an allele can be expressed using a letter. So let's take a look at both homozygous and heterozygous alleles. I want to use an example of freckles. So freckles is found on a dominant allele, which means that if a person has the dominant allele for freckles, they will have freckles on their body. So we characterize these alleles by letters. We can use the capital letter F. That means that is the dominant allele. And that capital letter F, that dominant allele, could have come from the paternal or from the maternal gene. A lowercase f means a recessive allele. And again, that could be on the paternal or the maternal chromosome. So let's look at homozygous chromosomes then. Homozygous chromosomes means that both the alleles are dominant or both the alleles are recessive. If both of the alleles are dominant, we would just write this as two capital letter Fs. That means that both the paternal and the maternal chromosomes carry the dominant allele. In this case, the person would definitely have freckles. If both of the alleles were recessive, we would use lowercase f's, meaning that the paternal and the maternal chromosomes both carried recessive alleles for freckles. That would mean that the person would not have freckles. In a heterozygous expression, that means that there's both a dominant and a recessive allele. So that means that one of the chromosomes would have the dominant allele and the other chromosome would have a recessive allele. So maybe the paternal chromosome had the dominant allele and the maternal chromosome had the recessive allele. In this case, the dominant allele is always expressed over the recessive allele. So this person would still show the physical characteristics of this allele, meaning they would have freckles. In polygenic inheritance, which we said was a phenotypic trait determined by interactions among several genes, the phenotype would be determined by the interaction between those corresponding alleles. For example, if a person has a gene for curly hair from their father, but a gene for straight hair from their mother, That person may have curly hair, they might have straight hair, or they might even have wavy hair depending on the relationship between those alleles. When one allele of a gene on a chromosome masks or overrides the effect of a different variant of the same gene on the other copy of the chromosome, we call that dominance. So in this example, in the case of freckles, in the heterozygous alleles, the dominant allele is the allele that expresses freckles, that we characterized by a capital letter F. This allele overrides the allele on the other copy of the chromosome. Dominance can be either strict dominance, codominance, or incomplete dominance. In strict dominance, an allele that is dominant will be expressed in the phenotype. That was the example that I used for freckles. The allele for freckles is dominant, so that if that allele is present on one chromosome, regardless of what's on the other allele on the other chromosome, that person will have freckles. So whether that person has two capital letter F's or a capital letter F and a lowercase f, in either case, that person would have freckles. The recessive allele would only be expressed if the same allele was present on both chromosomes of the homologous pair. In other words, the person would not have freckles only if both alleles on each of the copies of the chromosomes had the lowercase f or had the recessive alleles. So let's look at some examples of dominant and recessive traits. A dominant trait is an inherited characteristic that appears in an offspring if it's contributed from a parent through dominant alleles. Some examples of dominant traits, like I said, was freckles. So if the capital letter F is present, that person will have freckles. Another example is normal skin pigmentation, nearsightedness, farsightedness, astigmatism, free earlobes, the ability to roll your tongue, the Rh factor, type A or type B blood, and Huntington's disease. In recessive traits, which means that in order for this to be expressed, both of the chromosomes must carry the recessive alleles. Examples of recessive traits include albino pigmentation of skin, the absence of freckles, normal vision, attached earlobes, the inability to roll the tongue. If the Rh factor is absent, meaning a person is Rh negative, type O blood, sickle cell disease, cystic fibrosis, Tay-Sachs disease, and PKU. In strict dominance, an allele that is dominant will be expressed in the phenotype. However, there's also codominance. In codominance, both alleles are expressed in the phenotype. An example of codominance is type AB blood. To better understand type AB blood, let's first look at the different alleles. The dominant allele for type A is written as a capital letter I and a superscript A. The dominant allele for type B is a capital letter I and a superscript B. The recessive allele is written as a lowercase I. For type AB blood, there is only one combination. A person with type AB blood would have the following alleles: capital letter I with superscript A and a capital letter I with a superscript B. In this case, two dominant alleles are expressed in the phenotype. Let's compare this with type A blood. Type A blood is an example of strict dominance. With type A blood, the alleles can either be homozygous or heterozygous. Type A blood that contains homozygous alleles would be written as capital I, superscript A, capital I, superscript A. A type A blood that contained heterozygous alleles would be written as capital I superscript A, lowercase I. Likewise, a person with type B blood can contain homozygous or heterozygous alleles. A person that has type B blood with homozygous alleles would be written as capital I superscript B, capital I superscript B. If type B blood contained heterozygous alleles, it would be written as capital I superscript B, lowercase I. Type O blood contains homozygous recessive alleles. It would be written as lowercase I, lowercase I. Since codominance is a presentation where both alleles are expressed in the phenotype, the only example here that is codominance is type AB blood. When predicting simple inheritance in future generations, a tool called the Punnett square can be used. Punnett squares can predict the characteristics of individuals on the basis of their parents' alleles. A combination of alleles are placed in small boxes. Homozygous dominant, heterozygous, and homozygous recessive can all be possible combinations of alleles. The alleles from the mother and the alleles of the father are placed in different axes. Let's take an example of a mother who is homozygous recessive and a father who is homozygous dominant. For this example, we'll just use the letter A as the allele. So the mother is homozygous recessive and the father is homozygous dominant. The letters are carried down to the closest box in either the row or the column. As we can see here, the combination of a homozygous recessive mother and a homozygous dominant father will give offspring that all have the genotype capital letter A, lowercase a. So 100% of the offspring are heterozygous. If the capital letter A was the allele for normal skin color, and the lowercase a was the allele for albinism, and we know that normal skin color is strict dominance, then all of the heterozygous offspring would show normal skin color. If we compare that to a mother who is homozygous recessive and the father is heterozygous, we will see a different inheritance. So again, we carry the letters down into the closest box. And we see with this combination of mother and father, we have 50% that are heterozygous and 50% that are homozygous recessive. The homozygous recessive offspring will show signs of albinism. The offspring that are heterozygous will have normal skin pigmentation, but will also carry the recessive allele. A Punnett square can also be used to draw conclusions about the identity and genotype of a parent. For example, a man that has the genotype that is homozygous dominant, they cannot be the father of an albino child. In polygenic inheritance, where the phenotypic traits determined by interactions among several genes, we can't use a simple Punnett square to predict the characteristics of individuals based on their parents' alleles. In polygenic inheritance, one gene might suppress the other gene, or one gene might be complementary to the other gene, especially if it's recessive. For example, the risk of developing hypertension and coronary artery disease are linked to polygenic inheritance. Hypertension and coronary artery disease require both the genetic susceptibility and the environmental influences. If a person has the dominant allele and is susceptible to genetic hypertension, they may still be able to prevent high blood pressure by controlling their diet and their fluid volume. And if a person is susceptible to genetic coronary artery disease, they could prevent it by lowering their blood serum cholesterol levels. These factors make tracking polygenic conditions difficult through successive generations. Sex-linked inheritance involves genes on the sex chromosomes. The sex chromosomes are the 23rd pair of chromosomes. Unlike the other 22 chromosomal pairs, the sex chromosomes are not identical in appearance or in gene content. There are two types of sex chromosomes, an X chromosome and a Y chromosome. The X chromosomes are larger and they have more genes than the Y chromosomes. The smaller Y chromosome carries the SRY gene, which is not found on the X chromosome, that specifies that the individual will be a male. The normal pair of sex chromosomes in males is XY. The normal pair of sex chromosomes in females is XX. All oocytes carry an X chromosome because females only have X chromosomes. Each sperm carries either a Y or an X chromosome because males have one of each and can pass either one during meiosis. On a Punnett square, the ratio of males to females should be 1:1. Worldwide statistics show that there are 107 males per 100 females. It's suggested that there are more males than females because a sperm with a Y chromosome is faster and smaller than the one with the X chromosome. The X chromosome also carries genes that affect somatic structures. Genes that are on the X chromosome but not on the Y chromosome are called X-linked. X-linked genes don't follow the pattern of alleles on autosomal chromosomes. An example of an X-linked gene is the inheritance of color blindness. The dominant allele is a capital C, and we place that on the X chromosome, so we have a X with a superscript capital C. This results in normal color vision. A recessive allele is the lowercase c on the X chromosome, so we would have an X with a superscript of a lowercase c, and this results in red-green color blindness. A woman with her two chromosomes can either be homozygous dominant, which is X with a superscript capital C, X superscript capital C, or she can be heterozygous, X superscript capital C, X superscript lowercase c, and still have normal vision. She will be able to distinguish reds from greens only if she carries two recessive alleles, which are the lowercase c. But a male has only one X chromosome and one Y, so what determines whether he has normal color vision or has red-green color blindness will be whether the one X chromosome he carries will have a superscript capital C dominant allele or an X superscript lowercase c recessive allele. Sons produced by a father with normal vision and a heterozygous mother have a 50% chance of being red-green colorblind. Because the mother is heterozygous and contains a recessive allele for color blindness, we say that she is a carrier because she doesn't exhibit color blindness, but she carries the recessive allele that she can pass on to her offspring. Any daughter from a father with normal vision and a heterozygous carrier will have normal vision. The recessive alleles on the X chromosomes produce the genetic disorders in males at a higher frequency than in females. This ends our first video on inheritance and genetics. The second video will be shorter. We'll just be talking about some mutations.